Hi, welcome to our Chapter 5 Membrane Structure and Function and Lab 6 Cells recap video. Let's get started. So what we see for our plasma membrane is that it is going to be made of phospholipids in a bilayer or two-part layer with our polar head regions facing out and into the cell, and the little tails being the middle area of that membrane. Then we have cholesterol molecules, which are in between that phospholipid to help give our cell some structure, but also to allow it to be flexible. And then there are membrane proteins that are either integral, so they're in the whole membrane, or they are surface level. And it's important to know that our membrane is selectively permeable. We have a bunch of different membrane proteins. We have channel proteins that go all the way through the membrane that allow ions to pass through. We've got carrier proteins that can move specific molecules or ions through the membrane. We have cell recognition proteins that act like a name tag that are going to allow the cell to identify itself. We have receptor proteins, which are shaped so that molecules can fit into it and allow for certain things to happen within the cell. We have enzymatic proteins that are able to speed up chemical reactions. And then we have junction proteins, which help stick our cells together. We also look at our membrane as semi-permeable, and then we want to know how things can get in and out of the cell. And so one thing that comes up in this chapter is concentration gradients, where we have a membrane and only certain things are allowed through, so that one side will have more than the other side of certain molecules, and then where do things move. And as a whole, if we do not need energy, don't use ATP, we say that it's passive and things will move down their concentration gradient or toward that concentration gradient. It's like rolling a ball down a hill, or this is a wedge with these little silver dots. Same kind of idea there, moving down or toward that concentration gradient. If we have movement that does require ATP energy, that is going against, or where there's less, up that concentration gradient, and again requires energy. So let's start with the passive transports. That's going to include our osmosis and diffusion, and includes facilitated diffusion that uses a protein to move the molecules. What we see again here is that diffusion is just the movement from this concentration gradient, where there's a lot, and spreading out away down the concentration gradient. Osmosis is the movement of the water, not just any molecule. That's here in this second beaker where we've got a membrane and the big mustard yellow molecules can't get through, but the water will move to then space out, spread out those yellow circles. And so we call that osmosis. With this idea that the water can move, but not necessarily the molecules with osmosis, we have this idea of tonicity. And there are three different ways that solutions can look compared to a cell when we're looking at tonicity. So in an isotonic solution, the concentration of solute, or the dissolved thing, and the water is the same. I personally remember IS, isotonic, is same. And when it is the same, there's really no net movement of water. Water is moving in and out of the cell. In a hypotonic solution, there is going to be more stuff inside the cell, less water inside the cell. And so the water is going to move into the cell. And what we see is if it's an animal cell, that it can burst or lyse. And if it's a plant cell, then it gets a high turgor pressure. It's very happy that way. I like to remember hypotonic with the game Hungry Hungry Hippos. That hippos kind of looks like hypo, and I think about the game Hungry Hungry Hippos where you gobble up all the marbles in the same way your cells are gobbling up all of the water. And then, of course, we've got our hypertonic solution as our third option, where the water is going to leave the cell because there is more water in the cell than the solution. Our animal cells will crenate or shrink. Our plant cells will undergo plasmolysis, or the plasma membrane shrinks away from the cell wall. I remember the water moves outside of the cell by thinking about hyper kids going outside to run around at recess, just like the water goes outside of the cell. So how would you solve these problems? Well, the first thing is that you would be given a beaker or a solution and given a cell. You'll be told the amount of solute or salt or sugar or dissolved thing in the cell, and you'll be told the amount of dissolved thing in the water. And then you may have to do a little bit of math because the cell itself has to have 100% solution. So for example, if we say the cell has 5% salt, it has 95% water because then that whole cell is 100%. And the beaker itself has to have 100% solution. So same idea, if I say that the beaker has 25% salt, it has to have 75% water so that it could have 100% solution. So that's step one, is figuring out where is the water and where are my solutes right now, label them. Step two is that the water always moves from the high concentration to the low concentration. In my example, where there's 95% water to 75% water, well, the water is in a higher concentration inside 95% in the cell, and it will move outside. And then step three is to name that solution based on where the water goes. If the water moves into the cell, it's like Hungry Hungry Hippos, it's hypotonic. If the water moves out, it's like hyper kids going outside at recess, and it's hypertonic. If it is the same, that those numbers you've written down from calculating solute and water now are the same, it is isotonic. And if you've been following along with my videos, you know that there's a bonus point opportunity for you. And this is our practice. So we've got these beakers, beaker A, beaker B, and beaker C. And I want to know, where does the water move, into or out of the cell? And then can you name that solution, hypotonic, hypertonic, or isotonic? So feel free to pause the video here if you need to. And you will maybe need to do a little bit of math for cell B because I did not give you the water. But remember, it must equal 100% solution in the cell and out of the cell. And have some fun with it. And if you need some help, please let me know. When you have your answers for A, B, and C, feel free to email them to me. All right, moving then along, we have active transport, which requires ATP to be able to happen. So now we're not going with the concentration gradient, we're actually going against the concentration gradient. We have two major classes, exocytosis, which is like exit, so it is exiting the cell, and then we have endocytosis. endo, like enter. So we are bringing things into the cell. We have phagocytosis, which is bringing solids in. We have pinocytosis, which is bringing in liquids. I like to think of Pinot, like Pinot Grigio wine drinking, and that allows me to remember it is bringing liquid into the cell. And then we have a bulk version of this where I need to have a receptor mediated, meaning a receptor will be on the plasma membrane and I only bring in when I have enough. Switching gear to lab 6, we should feel well and good about our cell parts after our last chapter's lecture. But you will be identifying prokaryotic cells, including bacteria and archaea, looking at their glycocalyx, looking at their peptidoglycan cell membrane, looking at their nucleoid region with their one chromosome of DNA, looking at plasmids and pili and fimbriae and flagella, identifying the different organelles of prokaryotic cells. Then you'll move to eukaryotic cells, identifying nucleus for both plant and animal, nucleolus, endoplasmic reticulum, smooth and rough versions, ribosomes, mitochondria, Golgi apparatus, to name a few. You'll also look at cell wall versus the plasma membrane and centrioles and chloroplasts. Taking a look at our schedule, we see that today is chapter 5. There is a lecture quiz over chapters 4 and 5. Then there is the lab 6 virtual labs and the unit 2 discussion board. Anything that you don't get completed today can move to Friday as well. Then I like to remind us that sort of Saturday, Sunday, Monday are our catch-up mustard and mayo days, so you can catch up on any week 1 and week 2 smart books or virtual lab modules, and also the week 3 things that happened earlier. Your must-dos, that is our week 3 work, which is chapters 4 and 5, labs 5 and 6, and then our discussion board posts for unit 2. And then if you're looking for some things you may want to do or look ahead toward, there is the Chapter 6 Smart Book, there is the Lab 7 Virtual Labs, and of course, we were going to find ourselves in exam 2 time. And as we're looking forward to Tuesday, that's really where we're at, is the Chapter 6, Lab 7, and beginning to review for exam 2. All right, a nice short and sweet video. Thanks for watching. And as always, if you have any questions, please let me know. All right, have a great day.